there's a little bit of there's a double meaning in my title, Past, Present, and Future, because I'm going to talk a little bit about my students as well as our emphasis on the past, present, and future. So a little bit about past students. As basically, we use the past to understand the present. We use the present to interpret the past. And overall, we'd like to have a sustainable future for us and a diversity of life. And what I tend to do is my goal, and as many of you know, I have a fairly large group of students that we try to provide opportunities for individuals to develop as scientists whose interests are consistent with that vision. Let's see. Here we go. We cross disciplinary lines. When I was an undergrad, as a sophomore, I took Fundamentals of Ecology from the Odum textbook and fell in love with ecology, and I went to graduate school and learned that oceanography was just ecology on steroids as far as I was concerned. And you know, I mean, I was just eating up what Gary had to say this morning and what Steve and everybody's talks today. I just love it because I just, you know, I'm a natural historian and I just love what we're doing, what all of us are doing. But and certainly over the last 40 years, I've had a really great time doing some of these things. I'm interested in carbonate production and carbonate depositional environments. If you were here for Al's talk this morning, some of that ancient stuff, the stuff that's down below us, as well as the stuff that's being produced today. We use foraminifera as our model system for understanding a variety of things, from algal symbiosis and calcification to the Reef Indicators Lab is based on we've been developing bioindicators that are now being used on the Great Barrier Reef, for example, and bioindicator protocols that are now being used on the Great Barrier Reef. And about once or twice a year, I write papers with international colleagues interpreting geologic history aspects of usually the Cenozoic. And so this is, there was a discussion midweek about, you know, well, are our students doing interesting, you know, are they getting jobs or are they getting, doing interesting things? Well, it's coming out as blue and green, but there's no difference between those, I don't think. But the blue and greens are my former master's students who've gone on for PhDs, some of them. This isn't a comprehensive list. But if you look at the top, Kelly Boomer Donnelly's over here at the NOAA Southeast Region office in Grants, Beth Cucky Burkhardt's at the Bureau of Ocean Energy Management. I've got small business owners. I've got resource managers for the National Park Service. I've got quite a network of former master's students out there. So there are some really cool things that you can do with a master's degree. And if you look at this last line, and that's why I said the past, present, and future here, is that I think there are 5 graduate students here right now that are working on PhDs that with a variety of faculty, actually. So, my former PhD students aren't doing badly either. They're doing some cool things too. But I just wanted to brag a little bit about my master's students, since that's one of the questions that seems to come up. My current students are working on things ranging from the biology of forams to improving paleoceanographic proxies, and a number of students are working on actual reef building, key species of reef building corals and issues associated with corals, not forams. And thanks to the retirement of Norm Blake and other people actually, and my acquisition of several bivalve related students, Mike Drexler, Bridget McCrickard, and some others. We've expanded into the oyster realm, or not just the oysters, but in the bivalve realm. So here's our accomplishments for 2012. There were three master's theses, one on oysters, one on lobsters, and one on forams. So we span a range of things. These are some of our highlights for 2012. Adrian George got several accolades. Mike Martinez was selected to be a micropaleontologist on IODP Expedition 340. Natasha won a Cushman Foundation Research Award. Elizabeth Brown, the Gold Ring Award, which is awarded to a outstanding young female paleontologist, and we have a variety of endowed fellowships. Oh, and I forgot, although Julie was listed on, no, Julie wasn't listed, she was listed in the former students, but Julie Galkowitz, who's a Naus Fellow, essentially she's at the end of her Naus Fellowship right now, won the one of the outstanding dissertation awards. I think there were 2 or 3 were given from all USF for last year. So we seem to do okay. This is what I'm hoping for. I'm hoping to attend several commencement, or at least one commencement ceremony in the spring, but probably it'll be more than one. But these are who I'm hoping will graduate in the very near future. And Nolan was one of my adoptees from the Blake group. And I don't have a very good picture of him, so I went on the web and found found pictures of what he's working on. And how am I doing this? Mike Martinez has promised me he's finishing up, but he's got to get finished because to get his full-fledged faculty position over here at USF St. Pete. But he's working on forams as bioindicators of heavy metals in estuaries in Puerto Rico, and he had some NOAA and Sea Grant funding to do those projects. And Paul Supernand is started in my lab and then got interested in pteropods and has kind of wandered around. I'm still his co-advisor. Ben Flower was his principal advisor for a while, and Jose took over as principal advisor, but Paul is still involved in my group as well. So my latest adoptee is Elizabeth, and during her thesis research she observed some distinct morphotypes in one of the common foram proxies, and she has found that there are chemical as well as physical differences that were not previously reported from the Gulf of Mexico. When I was in Bremen and Bremerhaven, Germany, at a workshop or at a meeting that I have to go to each year for 3 years, I was talking with 2 different groups there who are very, very interested in collaborating and have invited Elizabeth to work. And we've applied for funding to do that, so we're hoping she gets to go. We're hoping not to see Elizabeth next year, that she'll be in Germany, if all goes well. Adrian does just amazing things. She was on the Oikos in May of last year. Then she got this NSF EPSI fellowship to work in Taiwan for the summer, and is hoping to go to China this coming year. And she's working on exploring the science and prevalence of coral diseases. She started out, she did her master's in collaboration with the Florida Aquarium. And this is our this is my highlight in all the highlights. I mean, we've got a really great group. But Ben, during his master's thesis, looking at essentially, we were looking at how the ingredients of the Corexit influence forams, and he discovered that they go dormant or go into a diapause type state when exposed to kind of an intermediate level. You can kill them outright if you give them too much. If you don't give them quite enough, they don't do much, they don't pay too much attention, although they will eventually bleach more readily than, but if you give them kind of an intermediate amount, they just withdraw and go into a sort of a diapause. And this has not been previously reported. And if you think about forams as being, the kind of #1 proxy used in oceanography, especially in the geosciences, but really, and there are only 10,000 or so extant species, as well as a million or so fossil species. It's to have discovered something like this is pretty amazing. So we're really excited about exploring this in his dissertation research. Natasha is doing some really cool things with photic stress in the diatom symbionts, and it's sort of ironic that the tool she's using is called a PAM fluorometer, but that's her baby there. she's carrying and she's skipping through the water, I guess. And as I said, the Joseph Cushman, the Cushman Foundation is the International Foundation for Foraminiferal Research, and she won one of the research awards. They give 3 to students from around the world each year. Libby Tyner has joined us as well. She was a Blake student. I was on her master's thesis committee, and she works full-time at SRI, interested in biofilms, and I've got her interested in how come the forams, when they start getting sick, start getting chewed upon by things, and when they're healthy, they look real pretty and almost nothing will be on their shell if you look at them under the SEM. And this individual was actually alive when we dropped it in some distilled water and put it on an SEM stub and looked at it under the SEM. And what's happened here is that the shell is weakened and the whole outer row of chambers has been chewed off by something. Basically, my whole lab studied foram bleaching throughout the 1990s, and we've been working on it off and on. And that's also related to the photic stress issue that are very much involved with the photic stress issue that Natasha's working on. So Katie Wirt is, her dissertation is in collaboration with Florida Fish and Wildlife. She's working with the GIS team over there, updating the critical habitat maps for the Acropora species, and as a prototype, because a number of other species are have just recently been listed, or in the process of being listed as threatened, and so these habitat maps that she's developing and testing and risk assessment, or not risk assessment, what's the term? anyway, she's trying to decide, how much effort is worthwhile in terms of the degree of upgrading these, or creating these additional layers to target your management strategies. Her master's thesis paper has just been, was accepted in December for applied geography, so she's got her first peer-reviewed publication. In another FWRI, Lucy Sprung-Bartlett she's working with the coral team, and she's looking at the stony coral versus octocoral recruitment. It's part of the phase shift or community shift that's going on in the reefs in the Florida Keys as the corals have died, as the stony corals have died back. The octocorals seem to be proliferating, and that's specifically the hypothesis that Lucy is testing on patch reefs that showed substantial dieback from the 2010 cold event versus ones that didn't show much die-off during the 2010 cold event. And she's looking at a series of patch reefs and comparing the octocoral and stony coral recruitment. Chris Netchie, another full-time FWRI and reef person, or coral person, and a part-time student is Chris is a taxonomist extraordinaire, and she is working with the museum specimens. She was with the museum collection. She's recently moved to the coral team, or is in the process of moving to the coral team. And she wants to compare the historical records around Florida with the modern coral survey data to see if the coral die-offs can be are being manifested in or how they're being manifested in diversity losses. And among my master students are Butch Ringelspa, who is the curator for exhibits at the Pier Aquarium. He's designed and constructed aquaria that are over in the in KRC that they're custom designed systems to do ocean acidification experiments using corals, particularly Acropora species. And last but not least is Heidi Toomey, who is also using chlorophyll fluorescence. The difference is that she's working on the chlorophyte-bearing forams as opposed to the diatom-bearing. The ones we have spent the most effort on are the diatom-bearing forms. But there's a whole group of chlorophyte-bearing foraminifera that have had very, very little biological work done on them. And Heidi's an ARCS fellow and a really terrific diver, and hopes to defend this semester. In fact, I strongly anticipate that she will be graduating this semester. So what's next? Where do we want to go? Well, if I wasn't here, I'd be working on my research proposals to I'm doing a proposal to NSF that's due next week to explore Ben Ross's discovery of dormancy in forams. That's a critical thing to be. We just got to find some funding to support that project, because it's just I think that's it should be not mine, Ben's science or nature paper. And then I've got a couple of dream projects that most of the time I just advise students and go to meetings. But if I was going to dream about what I could do, there are two things that, one would be The diatoms are produce lipids as their storage product, and tend to be lipid rich, and symbionts are designed, if you will, or to excrete most of their photosynthate. So most of what they fix, they're designed to excrete to their host. And in the host suppresses their frustule formation as well. So they should have extremely good potential, or at least with genetic engineering, maybe some of their qualities could be put into some other kinds of algae that would allow essentially photosynthetic production without a lot of photosynthetic growth, or without a lot of algal growth. And that's the key thing, because they're designed to excrete their primary production, not to grow with their primary production. Well, they'll grow if you give them plenty of nutrients, but nutrient starved. They're designed just to photosynthesize and excrete their photosynthate to their host. And a completely different project, but one that's tied into my more geologic interests, is the whole issue of what we call them the mesophotic and oligophotic systems that are right near the uppermost thermocline, nutricline, and with internal wave events, they can, this terrific food source, if you will, can flush right up onto these shelf margin kind of, we're talking again 60 to 90 or so meter deep systems that are in such clear water that the light penetrates that deep. Pulley Ridge is an example here on the West Florida shelf. And there's got to be some interesting pumping of more CO2 lower or more CO2 rich waters that are in the nutricline, or in the aphotic zone, or below the photic zone, that then when it's pumped into shallower water, that may account for submarine cementation that occurs, that oftentimes, at least especially in the fossil record, these shelf margin kinds of reefs tend to be absolutely full of submarine cementation. And so, so that's kind of, this is my dream for helping there be a future for all of us. And this one's my dream for how to bring all my interests in biology, geology, chemistry, and physical processes into understanding these because this is the environment that the larger forams, I didn't even mention that, but the larger benthic forams that are a lot of those limestones that Al Hine was talking about, that oil is found in, lived in exactly these kinds of environments from about 50 million years ago until about, well, they still live in those environments, but they were most prolific from about 50 or so million years ago to about 15 or so million years ago, and a lot of oil and gas comes out of rocks of that age that are foram limestones. Also, where they're not foram limestones, they are, if they're shallower, they're aquifers. And one of the major ones that we are all familiar with, because when we turn on the water fountain, is the Floridan aquifer, because it's foram. Forams are very, very abundant in the limestones that make up the Floridan aquifer. So anyway, you've got a little bit of an idea of what we all do and what I dream of, and that's that's about all I have to say. So if anybody has any questions, I guess there's Dean Dixon. And I think I'm since Mark went a little over, I think I wasn't too much over.
Yeah, you're not over. You're 5 minutes under.
Oh, okay. Bob had a question.
What are the certain chemicals in Dan's research?
Oh, what were the chemicals? He actually, the two chemicals that he used were both alcohol. I just went blank. Propylene glycol was the not, and it's in hand lotion. You know, I mean, it's a very common chemical. And the other one was, what was the? Butoxyethanol. Butoxyethanol. Oh, right. And the thing was is that we thought, because the initial experiments were with propylene glycol, maybe it was just because it was such a benign chemical. But then he used the butoxyethanol, which is at least an order of magnitude more toxic, and they did the same thing. It just did the concentrations that caused them to go dormant was an order of magnitude less. Thank you.
We have time for one or two questions.
Any other questions?
No, let's.
Did you take a special course in time management?
Actually, I did years ago. You mean to have that many students? Actually, Jose, Gabe Vargo, Ted Van Vliet, and myself, and I think Al Hine too, have all, and I'm sure Bob too, over the years have all been on at least 100, something like 100 committees in our long careers. And so and about half of the committees I've been on were my own students, and the other half were other people's students. But you know, one of the things is that I really kind of emphasize, we've de-emphasized master's students to some degree. The university wants us to de-emphasize, and yet you know, I really, you know, and some people say, well, that's what everybody's good at master's students. Well, I'm not so sure that's true, having rescued a few over the years. But I really am. That is what I'm best at, you know, because maybe I mother the PhD students too much. I don't know. You can ask them. But I do have quite a few PhD students right now, and not so many masters. Any other questions?
There's no more. Let's thank Dr.
2 years ago, the interview was all about my extensive academic experience, and I had to give a talk about the different academic qualities that they would judge me on. And frankly, nobody here has any idea of what I do. So I'm really in favor of doing these talks. Now, what I do is really, it belongs in a marine institute because I study submarine volcanoes, and I'm looking at the links between subduction and ocean island volcanism. But nobody in this college does any research anywhere near what I do. So my closest colleague at USF is Jeff Ryan, who's chair of the geology department. Given that, I'm going to start out with background. And hopefully in the beginning, it's like all familiar with what you learn in geological oceanography. And then I'm going to start getting into more detail, and I'm going to lose people. And then I'm going to get into the details of what I actually do. I'll probably lose most of you. And then I hope to bring you all back in at the end. So if I start to lose you, don't panic. And I have a couple of props with me. All right, so at most people, you're familiar with the water cycle, right? And when we talk about geochemical cycling, we're talking about reservoirs where the mass of your element of interest resides. We're talking about fractionations between reservoirs. the processes which cause those fractionations. So what's a fractionation? In stable isotopes, it's you're separating the light isotope from the heavy isotope for reasons of temperature or chemical reaction. If we're talking about radiogenic isotopes, we're talking about trace element partitioning, which pulls apart the parent and daughters. And then if you let those reservoirs age with time, if they are isolated, then you can get different geochemical signatures, develop a unique isotopic fingerprint or a unique stable isotopic fingerprint. So in the case of the water cycle on the surface of the Earth, our reservoirs are surface water, lakes, rivers, and ocean, and the atmosphere. One of the important processes which fractionates elements is evaporation and precipitation. During that fractionation, the light isotope prefers to be in the atmosphere, leaving the heavy isotope behind. And then when you rain, you're raining out the heavy isotope. And so with time, you develop a reservoir of ice that is light, which adds and subtracts from the oceans, giving us stable isotopic variations in the composition of the ocean as a function of the volume of different reservoirs, including ice. So everyone's with me so far, right? Okay, good. So this is my water cycle. And instead of time scales of days, months, years, decade, millennial, my time scales is billions of years. All right. My reservoirs include rocks in the crust, and that's oceanic crust and continental crust, the mantle, and my processes of fractionation are the generation of melt. And so I'm interested in what's the water cycle, what's the long time understanding evolution of water and carbon in the mantle. So the stuff coming out, the flux out, are the magmas. Mid-ocean ridge volcanism, oceanic island volcanism, arc volcanism. The flux into the reservoir is what goes back down, what gets stuffed back down by the subduction process. And understanding the fractionations that happen during melting and during subduction will allow us to identify different reservoirs that have different geochemical fingerprints that evolve in time to give us characteristic radiogenic isotopic fingerprints. Are we good so far? Okay. So, my main research is what is the carbon and water concentration in the mantle? How does it evolve with time? Who gives a shit? Isn't this all esoteric? Well, actually, without plate tectonics, you don't have life. Volcanoes, water, those are the basic building blocks in a planetary sense for the evolution of life. Without water in the mantle, things would be too viscous because add just a little bit of water, you greatly reduce the physical properties of the solid. So the viscosity decreases, electrical conductivity goes up, and so because you have water in the mantle, you can have plate tectonics, and so you can have life, and then you can have coral reefs and sediments and climates and everything else. So it is a fundamental, and if you're a paleoclimate person, it is that flux of CO2 that is the background flux of carbon into the atmosphere. So if you want to know about ore deposits, you want to know about volcanism, you want to know will volcanoes explode or not explode? Should you live next to this volcano? Should you not live next to this volcano? Why do we have plate tectonics? Why do planets have life? That's my research feeds into that. But mostly it's more esoteric. Okay. So the major fractionations that I am concerned about are melting, which is the flux out of the mantle. And during that melting, and I'll go into a little bit more detail, certain elements get concentrated in the melts, certain elements preferentially get left behind, and that leaves us a chemical fingerprint of the mantle that has melted or has had melt added. And so the partitioning of interest is when you have a silicate melt in equilibrium with a mantle assemblage. What is the equilibrium in terms of the partitioning of elements? And okay, geological oceanography class quiz. The mantle is made out of what? David?
Prototype.
Thank you. Okay. Peridotite is made up of?
Olivine, amphiboles.
No. If there's water. What's an amphibole without water?
Kerosene.
Pyroxene. Good. So it's mostly olivine, pyroxene, which comes in two flavors. The flavor that has calcium is clinopyroxene because it's big and it torques the structure. The flavor without calcium is orthopyroxene. Okay. So we're looking at the why do we get melting and how do elements partition when you get melting, and then that goes up and becomes the crust, or what I refer to as the scum. Okay. And then we generate oceanic crust, and then we let it interact, and it's fairly dry. We let that interact with seawater. As plate tectonics moves the oceanic crust across the seafloor inexorably towards the subduction zone, it's sucking up water. You think about that oceanic crust, and what's the oceanic crust made out of, David?
Basalt.
Basalt, okay. So that's when you melt the mantle, you get a basaltic magma, it turns into basalt or gabbro. Felt relatively dry, it's a sponge, it's sucking up water, sucking up water, sucking up water. You add water to olivine, what do you get? Somebody else other than David? Serpentine, it can hold up to 11% water. And then you're going to take that now wet sponge to the subduction zone, and then you're going to stuff, and it has sediments on it, combination of all the different pelagic and carbonate and all sorts of things. Okay, it's got sediments on it. And then you're going to stuff it back down. So we have stuff that crystallized from hot magma. It sucked up water, and now we're going to stuff it back down into the garbage chute called subduction zones, and then we're going to dehydrate it. So this oceanic crust at very different pressures and temperatures, different lithologic components in that package of the subducting oceanic lithosphere will dehydrate. During that dehydration, we have equilibrium between the rock and a hydrous fluid. Elements partition differently than they do when the equilibrium is between a silicate melt and a silicate rock. And we'll go into what those differences are. Once you generate a chemical fractionation by a process that you understand, and you keep it isolated and you let it evolve, you can evolve into different radiogenic isotopic compositions that you can then use as a tracer. The big difference, and when you're a biologist, you talk about conservative tracers and non-conservative tracers. So a biologist, raise your hand, what's the difference between a conservative tracer and a non-conservative tracer? Oh, come on. Jose.
Kind of what sort of a tracer you were looking at.
Give me, just, in some tracers.
I'm not a biologist, but I'd
Okay.
say one's conservative and the other one's not conservative.
Right. Excellent answer. Excellent answer.
That's actually the physics question, not biology.
Okay, so
Conservative are good ones, non-conservative, biology mucks up.
that's right. The biology mucks it up. They're involved in the process. And so the conservative tracers are one that are not modified by the various processes that are going on. The non-conservative tracers are the ones that are modified by various processes that are going on. So you have to understand the process that caused the fractionation, and then the process as the system is evolving that then changes those tracers.
Different one satisfies conservation equation and another one doesn't.
Another excellent answer. Okay. Volatiles are a little different, and I gave you a hint because, oh, you got it unwrapped. Okay, good. If you add a little bit of water, you're going to be able to generate a lot more melt. And clearly the amount of water and what minerals it's in controls the dehydration process. So understanding the volatiles is not only as volatiles as a tracer, but volatiles as an active participant and control on the processes. So it's doubly important to understand their variation. And I do my work on mid-ocean ridge basalts and oceanic islands, and I get to go to lovely places. All right, so class project. Where do volcanoes occur? Steve. Okay, mid-ocean ridges. hot spots and subduction zones. Okay, which tectonic environment has volcanoes that tend to explode? Subduction zones. Okay, subduction zones are where most of the hazards are. Okay, so why do we have volcanoes in these different tectonics environments? Okay, this to me is a basic end of the first year. You've had geological oceanography comps question. Maybe not so much here, but everywhere else would have been. Why does melting occur? Okay, there's three ways you can get melting of the mantle. You can heat it up, you can raise it up, or you can add water. And so normally you think if you've got a solid, it's a solid, I want to melt it, what am I going to do? I'm going to put a torch under it, I'm going to heat it up, and it will melt. That does occur when you are initiating a new rift, and you've got mantle coming up and heating the lower continental lithospheric mantle. That's a way that the rock stayed there, the temperature changed, and you can get melting. The most common way to get melting, you can think about the melting, okay, the solidus. Now does this have a laser pointer? Is that, yeah, okay. So the solidus is the temperature at which melting begins. For most materials, if you squeeze it more, you have to add more heat to make it melt. There are two materials on the surface of the Earth that don't follow that rule. What are they? Water is the most important for us. It's why we can freeze the tops of lakes, and we still have life in our freshwater lakes. The other is menthol. So if you want to do experiments, you can do experiments with menthol. But water is a real exception. So for most materials in geology, oops, okay, get this right, there we go. Here's the solidus. So this is pressure. So this is going down deeper in the earth. So pressure is increasing as we're going down. This is temperature. Temperature is increasing as we move to the right. This is the melting curve of the solidus for mantle materials, peridotite. And the more you squeeze it, the more heat you have to add to make it melt. And so let's do mid-ocean ridges and hotspots. During mantle convection, the mantle convects adiabatically. What's adiabatically? You're the physicist.
Doesn't exchange heat.
Doesn't exchange heat. So when we have our mantle convection, which is driving plate tectonics, the mantle, solid mantle, is rising up. It's rising up quickly enough that it's not thermally equilibrating. And so you've got hot mantle rising up. Eventually, at some point...
How quickly is it quicker? What is the time?
Well, the mantle convection is occurring at the same rate as plate tectonics. And so that's centimeters per year, the rate your fingernails are growing. So at some point, this hot mantle is rising up, it intersects the solidus, and you get melting. Underneath mid-ocean ridges, that's typically 30 to 10 kilometers depth. Oceanic islands are coming from deeper, a little hotter. They start melting maybe about 100 kilometers depth. And so this is called adiabatic pressure release melting, adiabatic melting. And this is the main mechanism for getting melting to create new oceanic crust and oceanic islands. And so this is, you had a geotherm, you started rifting, you're heating it up, and so you shift the temperature over. And then this one is really important. Just add a little water, and when you add water, you greatly reduce the melting temperature. And so you can have a rock in the mantle wedge above a subduction zone. It's not at the melting temperature. You add water to it, and then it will melt. Okay? So you can raise it up, you can heat it up, or you can add water and change the melting temperature. Those are the three ways to get melt. When you melt, the things that control the partitioning of trace elements into mantle melts, or basaltic melts, are the ionic charge and the size. And so it's simply a function of these mantle minerals. Olivine and pyroxene have a fairly tight mineralogic structure, and they don't, there's a little bit of Dixon's rule of geochemistry. There's a little bit of everything and anything. But in the mantle, when you melt it, if you are large or you are highly charged, you would prefer to be in a more relaxed environment of a silicate melt. And so those elements that, when given a choice, would rather be in the easy chair than the tightly constricted chair, those elements are called incompatible. They are incompatible with mantle mineral structure. Those elements that are quite happy sitting in these tightly constrained chairs are called compatible elements. So when you melt the mantle, the incompatible elements go off in the melt, the compatible elements stay behind. That's a chemical fractionation. And so that's why we get, and you form the oceanic crust, and then eventually it turns into continental crust. And that's why the continental crust has higher concentrations of our heat-producing elements like uranium and lead and thorium. And so when you're talking about eroding off of continental crusts and then it turns into sediments, all these things, is a different reservoir, because a long time ago we created the crust. It was concentrated in incompatible elements, and those are the things that then you are reflected in the sediments. So incompatible and compatible. If you ever have to suffer through a talk in a session at AGU that I would be in, you are expected, if you are an igneous petrologist, you know every element, you know the periodic table, you know its distribution coefficient, how it partitions between minerals and melts, and you know the various ratios. So if two elements have the same distribution coefficient, then they are not fractionated by the melting process. So that ratio should be a canonical ratio. It won't change with this fractionation process. If they have greatly different distribution coefficients, the way they partition, then you can see changes in very different ratios in the melt crust versus the residual mantle left behind. You still with me? Okay. Mostly. I've lost a few. Okay. But so it's very annoying when you go to a talk in one of my sessions, because you are expected to know every element, every ratio has a meaning, and nobody explains it anymore. So here's an example. So here's our mantle. We formed a melt. That melt is going to go up and form the crust. Here's a sequence of elements. We love the rare earth elements. I won't go into details as to why. But this is more incompatible. This is more compatible. And so the mantle left behind after melt extraction is depleted in incompatible elements. That's the chemical fingerprint of depleted mantle, which is now melting to give us mid-ocean ridge basalt. The continental crust is complementary in its trace element patterns. It is enriched in incompatible elements, depleted in compatible elements. When we look at elements that will be important in radiogenic isotopic fingerprint evolution, the parent in rubidium strontium, the parent prefers the crust, the daughter prefers the mantle. In neodymium samarium, it's reversed. Neodymium prefers the mantle, samarium prefers the crust. And so that leads us to characteristic isotopic evolution for these different reservoirs created through the process of mantle melting. And then this just shows various patterns for incompatible element enriched continental crust, incompatible element depleted mid-ocean ridge basalt, which is derived from a second melting of a previously depleted mantle. You're getting my igneous petrology course all in one 15-minute interval. And these are the elements that we love to play with in terms of radioactive decay schemes: rubidium strontium, samarium neodymium, uranium lead, thorium lead, with half-lives all either greater than or close to the age of the Earth. And so they decay away, and all of you should know, how many half-lives do you see a good signal if we're talking about radioactive decay? 5, 6, 4, 5. And so we are clearly, if we have a fractionation caused by melting or caused by subduction, we're clearly in the time frame for these that we can see changes in evolution of the isotopic composition. And here we have the same thing And so because we have